AI is now designing drugs, and it just saved a baby's life. In August 2024, a baby boy was born in Philadelphia. His name was KJ Muldoon Jr. His family called him KJ. Within days of his birth, the doctors at Children's Hospital of Philadelphia ran genetic tests and found something that stopped them cold. KJ had been born with a condition called severe carbamoyl phosphate synthetase 1 deficiency, CPS1 deficiency. It's a genetic metabolic disorder that affects roughly 1 in every 1.3 million newborns. The gene responsible for producing a critical enzyme in the liver's ability to process protein and eliminate ammonia was broken in both copies. Without that enzyme working properly, ammonia builds up in the blood to toxic levels. If left untreated, it causes brain damage and death. The standard options for severe CPS1 deficiency are grim, a highly restrictive diet that limits protein so severely it stunts growth, medications that attempt to compensate for the enzyme the body can't produce, and eventually for many patients a liver transplant, which is a major surgery with serious risks in an infant, requiring lifelong immunosuppression afterward. There was no specific approved treatment for KJ's exact genetic variant of this disease. Doctors had essentially never seen it before in this precise form. What happened next changed medical history. The team at Children's Hospital of Philadelphia and Penn Medicine contacted researchers and immediately began a race against time. They had one goal: design a treatment that could correct the specific genetic error in KJ's liver cells, the exact mutation that was breaking the enzyme, before the disease caused irreversible damage. In September of 2024, they started. In late February of 2025, just 6 months later, they gave KJ the first dose of his treatment. To put that in context, the normal timeline from identifying a genetic treatment target to first human administration is measured in years, sometimes decades. The regulatory process alone typically takes years. Clinical trials, animal studies, safety reviews. The process is long by design because the stakes of getting it wrong are high. KJ's team did it in 6 months, and they couldn't have done it in 6 months without artificial intelligence. This video is about what happened, how it was possible, and what it means for every person watching who has a family member with a rare disease, a chronic condition, or any medical situation where the current treatments are inadequate. Let's start by explaining what they actually did in plain language, because the science is genuinely fascinating. The technology used to treat KJ is called CRISPR base editing. CRISPR is the gene editing tool you've probably heard about over the last several years. The basic idea is that CRISPR acts like a pair of molecular scissors that can be guided to a very specific location in the genome and make a precise cut, or in this case, a precise edit. Base editing is a more refined version of CRISPR that doesn't cut the DNA strand at all. Instead, it changes individual letters in the genetic code, swapping one chemical base for another, the way you'd use a word processor to correct a typo without deleting and rewriting the whole sentence. In KJ's case, the goal was to find the specific typo in his CPS1 gene, the exact genetic letter that was wrong, and change it to what it was supposed to be, in his liver cells, in his body, without taking cells out, editing them, and putting them back. The therapy goes in, finds the target, makes the correction, and the cell's own machinery takes it from there. The challenge is that every patient's specific mutation is different. The typo in KJ's gene is not the same typo as another patient's. You can't use a generic drug because there isn't a generic version of this problem. You have to design the treatment from scratch for that specific patient with that specific mutation. Here is where AI enters the story in a fundamental way. The process of designing a gene editing therapy requires analyzing an enormous amount of data. You need to identify the exact mutation. You need to design a guide sequence that will direct the base editor to precisely the right location in the genome. You need to predict whether that guide sequence might accidentally edit the wrong locations elsewhere in the genome, what researchers call off-target effects. You need to model how the therapy will be delivered into liver cells using what are called lipid nanoparticles, essentially tiny fat bubbles that carry the genetic payload into cells. You need to test all of this computationally before you ever go near an animal, let alone a human. Each of these steps, done by human researchers working manually through the literature and running their own experiments, would take months or years. AI systems that have been trained on the vast literature of genomics, protein structure, cell biology, and pharmacology can perform much of this analysis in hours or days. The researchers working on KJ's case used AI tools to rapidly scan for potential guide sequences, to predict off-target risks, to help design the lipid nanoparticle delivery system, and to synthesize and integrate findings from thousands of of relevant scientific papers that no human team could have read and processed in that time frame. The Broad Institute at MIT and Harvard, Jennifer Doudna's Innovative Genomics Institute at UC Berkeley, and researchers at multiple other institutions worked in parallel, sharing data and analysis in real time. AI tools for genomic analysis, protein structure prediction, and safety modeling ran continuously, turning what would have been years of sequential work into months of parallel work. By late February 2025, KJ received his first dose. A second and third dose followed in March and April. By April 2025, the results were clear. KJ was tolerating significantly more dietary protein than before. He needed less medication to control his ammonia levels. He had recovered from ordinary childhood respiratory infections, the kind of illness that would have been dangerously destabilizing before treatment without a dangerous buildup of ammonia. He was described by his medical team as growing well and thriving. In June 2025, after 307 days in the hospital, KJ went home. On December 8, 2025, Nature magazine named KJ Muldoon to its list of the top 10 people who helped shape science that year. Not a scientist, not a researcher, a baby, because what his case represented was so significant, so demonstrative of what is now possible, that it reshaped how the scientific community thinks about the relationship between AI and personalized medicine. Now let's zoom out from KJ's story and talk about what it means for the broader landscape of AI in medicine, because the implications are enormous and they go far beyond rare genetic diseases. For most of human history, developing a drug has been an extraordinarily slow and expensive process. The standard estimate is that bringing a new drug from initial discovery to FDA approval takes between 10 and 15 years and costs somewhere between $1 and $3 billion on average. And the failure rate is staggering. Most drugs that enter clinical trials never make it to approval. The majority fail for reasons related to efficacy or safety that weren't predictable from the early stage research. The reason the process takes so long and costs so much is fundamentally about information. Biology is almost incomprehensibly complex. The human body contains roughly 37 trillion cells, each with around 20,000 genes, each gene capable of producing multiple proteins, each protein capable of interacting with thousands of other proteins in ways that shift depending on cell type, age, health status, and countless other variables. Understanding how a drug is going to interact with all of that complexity is hard, really hard, and doing it mostly through sequential human-designed experiments that each take months to set up and analyze is the primary reason the timeline is so long. AI is attacking this problem from multiple angles simultaneously. The first is what AlphaFold did for protein structure prediction, because this is the foundation of much of what's come after. Proteins are the workhorses of biology. They carry out virtually every function in the body. Understanding what a protein does and how drugs might interact with it requires knowing its three-dimensional structure. Determining protein structures experimentally was one of the most time-consuming processes in biology. It could take a research lab years to determine the structure of a single protein. In 2020, DeepMind's AlphaFold AI system solved this problem with stunning accuracy. It can now predict the three-dimensional structure of a protein from its genetic sequence alone in minutes, with accuracy that matches experimental methods. AlphaFold has now predicted the structures of essentially all known proteins and made that database publicly available. Researchers all over the world are using it as a foundation for drug discovery research that would have been impossible before. The second angle is AI systems that can predict how potential drug compounds will interact with protein targets. Traditional drug discovery involves synthesizing and testing thousands or millions of compounds to find ones that interact with a target protein in the desired way. This is expensive, time-consuming, and still produces mostly failures. AI models trained on the vast database of known drug-protein interactions can now screen billions of virtual compounds computationally, predicting which ones are most likely to work before anyone makes them in a lab. This doesn't eliminate the need for laboratory work, but it dramatically narrows the field, focusing expensive lab resources on the most promising candidates. The third angle is AI-assisted clinical trial design and patient matching. One reason clinical trials fail is that the wrong patients are enrolled. A drug that works for a specific genetic subgroup of a disease might fail a clinical trial if that trial enrolled patients without that subgroup being properly identified. AI systems that can analyze patient genetic data, medical history, biomarkers, and other variables are getting better at identifying which patients are most likely to respond to a given treatment, potentially improving success rates significantly. The fourth angle, and perhaps the most profound, is what is being called the AI co-scientist. This goes beyond AI as a tool that runs calculations. An AI co-scientist is a system that can read scientific literature, generate new hypotheses based on what it reads, design experiments to test those hypotheses, interpret the results of those experiments, and iterate. It operates as an active participant in the research process, rather than just a powerful calculator. Google DeepMind has been developing systems specifically for this purpose. Anthropic has published research on using AI systems for scientific reasoning. Multiple pharmaceutical companies have AI co-scientist programs underway. The results so far are preliminary but genuinely exciting. In some domains, these systems are surfacing unexpected connections between established findings, connections that human researchers hadn't noticed because no individual person could read and synthesize the full body of literature in a given field. There is a disease called ALS, amyotrophic lateral sclerosis, better known as Lou Gehrig's disease. It's a progressive neurological disease for which there is currently no cure and very limited treatment. Researchers have been working on it for decades with limited success. AI systems analyzing the scientific literature on ALS have begun generating hypotheses about potential treatment mechanisms that are now being tested in the lab. None of them have produced an approved treatment yet, but the speed at which new hypotheses are being generated and tested has accelerated dramatically. The same is happening in cancer research, in cardiovascular disease, in Alzheimer's research, in infectious disease, in essentially every area of medicine where the bottleneck was the speed at which human researchers could process information and design experiments. AI is beginning to change the math. Now let's be honest about the challenges, because there are real ones, and responsible coverage of this topic requires acknowledging them. The AI tools being used in drug discovery are powerful, but they are not magic. They can predict protein structures with high accuracy, but predictions still need experimental validation. They can generate drug candidates computationally, but those candidates still fail at high rates when they reach biological testing. The history of medicine is littered with treatments that looked extraordinarily promising in computational models and failed in clinical trials because biology is more complex than any model, no matter how sophisticated. The timeline to turning an AI-assisted drug discovery breakthrough into an approved treatment that patients can actually access is still measured in years. Regulatory processes, clinical trials, manufacturing, distribution, all of these take time, and they take time for good reasons. The regulatory safeguards that protect patients from unsafe treatments don't disappear because the discovery process was faster. There are also serious questions about access and equity. KJ's personalized gene editing therapy reportedly cost in the range of $1 million. Treatments designed for one patient at a time using cutting-edge technology are inherently expensive. If the future of medicine is personalized treatments designed by AI for individual patients, who can afford them? How do insurance systems handle treatments that are designed for one person? These are not questions that technology alone can answer. And there are ethical questions about the boundary between treating disease and enhancing biology. If AI-assisted gene editing can correct the genetic errors that cause deadly diseases in infants, what's the line between treatment and enhancement? These conversations need to happen in public, involving more than just the researchers and companies developing the technology. With all of those honest qualifications in place, the direction of travel is clear, and it is genuinely remarkable. The time from identifying a disease-causing mutation to designing a therapy that corrects it has gone from decades to months. That is a compression of time so dramatic it should give everyone pause to consider what it means. For the roughly 300 million people worldwide living with rare diseases, most of whom have no good treatment options, the personalized medicine revolution powered by AI represents the most significant reason for hope in a generation. The doctor who worked on KJ's case said it publicly. Now we have to show that we can scale this from CRISPR for one to CRISPR for all. That is the challenge. That is the work being done right now in labs, in hospitals, in AI companies, and in regulatory agencies, working out how to evaluate treatments that have never existed before. KJ Muldoon went home in June 2025 after 307 days in the hospital because a team of researchers had access to AI tools that compressed years of work into months, and because they used that time to design something that had never existed before, a medicine made for one person, built in record time, that worked. He is now a toddler, growing, thriving, playing. That is what AI in medicine looks like when it works, and it is only the beginning. Subscribe if you want to follow the technologies that are quietly changing what's possible in human health, energy, transportation, and daily life. We cover this stuff before it goes mainstream, and we cover it honestly, every single week.